Hi there, welcome to this video. This one's all about carbohydrates. So carbohydrates have three main categories. So the first one is called a monosaccharide. The second one is called a disaccharide. And the third is called a polysaccharide. So look at the prefixes on the words. Mono means one. So monosaccharides are made of one sugar unit. Okay. Disaccharides, di means two. So these are made of two sugar units joined together. And thirdly, poly means many. So these are made of many sugar units joined together. Now, most of the sugar units that we need to know look like this. So that would be one for a monosaccharide. Two would join together like that for a disaccharide. And then many, would join together like this for a polysaccharide. And sometimes it has little branches coming out the sides as well. So what we're going to do is we're going to go through these three categories in turn. So first of all, let's look at monosaccharides. So the monosaccharide that you have to know from memory is glucose. And first of all, you need to know how to draw glucose. So it's a six-sided shape. So we draw it like that. And in the top right-hand corner, we've got oxygen. On the corners, we've got carbons. And all carbons have to have 4 bonds. Now, one of the carbons is slightly different. These two here, or two of them, sorry, are slightly different. These two here above one another are slightly different, so I'm going to leave those ones to last. The others all have a hydrogen atom above the carbon and a hydroxyl atom below the carbon. Hydrogen above, hydroxyl below. Hydrogen above, hydroxyl below. The oxygen only has 2 bonds and therefore there's no hydrogen or hydroxyl attached. These two that I've highlighted in purple are the opposite way around. So the hydrogen is at the bottom and the hydroxyl at the top. Hydrogen at the bottom. And the top one on the last one is different. It's actually CH2OH. Now I suggest you replay that part of the video again and again until you can draw that off by heart. Okay, it's really, really important that you can draw alpha glucose from memory. Now, this molecule of glucose, more specifically, is called alpha glucose. This is the symbol for alpha, alpha glucose. And you also need to know how you would draw beta glucose. So beta glucose is kind of similar, but a little bit different. It's carbon number 1 that's different. So if we draw glucose the same, so oxygen in the top right-hand corner, 6-sided shape, all the corners need carbon. We know that these two are different, so we leave those to last, and all the others are the same. But now I'm going to tell you that in beta glucose, this one is different as well. Okay, so ordinarily we'd say hydrogen at the top, hydroxyl at the bottom, but we'll miss that one out for now. So hydrogen at the top, hydroxyl at the bottom, hydrogen at the top, hydroxyl at the bottom. These two are the other way around. Hydrogen at the bottom, hydroxyl at the top, hydrogen at the bottom, and something slightly different on the last one, CH2OH. Now, the way that this beta glucose is different to alpha glucose is just simply that this hydrogen and hydroxyl group are the other way around. So the hydroxyl group's at the top on carbon 1 and the hydrogen is at the bottom. And that's what makes beta glucose different from alpha glucose. So you need to be able to describe those. The next thing you need to be able to do is number the carbons. So starting from oxygen, go clockwise. So this is carbon number 1. Then you see, are there any others attached? No. So go round the ring. This is carbon number 2. Are there any attached? No. Carbon number 3. Are there any attached? No. Carbon number 4. Are there any attached? No. Carbon number 5. Are there any attached? Yes. So that's carbon number 6. So same on here. It'll be carbon number 1, 2, 3, 4, 5, 6. So the way we describe the difference between alpha and beta glucose is we say that the hydrogen and the hydroxyl group on carbon 1 are reversed. They're the other way around, aren't they? So the hydrogen and the hydroxyl group on carbon number 1, this is a little 1, are reversed. That's the difference between alpha and beta glucose. The other thing about glucose molecules is that they have 6 carbons. And because they have 6 carbons, we call them hexose sugars. They're not hexagons. That would mean 6 sides. Well, they actually are hexagons as well by chance. But this word hexose means that they have 6 carbons. Okay, they're made of 6 carbons. The next thing we need to know is disaccharides. So let's go on to a new board. So a disaccharide means that. So a disaccharide means 2 sugar units joined together. So the first thing you need to do is you need to draw 2 alpha glucose molecules side by side. So remember, 6-sided shape, oxygen in the right-hand corner, carbons on the other corners. These 2 are different, but all the rest are the same. So add 4 bonds for all your carbons. Hydrogen, hydroxyl, hydrogen, hydroxyl. Careful, when you draw your hydroxyl, it should be the oxygen that's joined to the bond and the H next to it. These 2 are the other way around, so hydrogen on the bottom, hydroxyl on the top, hydrogen on the bottom, and different one on the top, CH2OH. Then you need to draw another one next to it, 6-sided shape. Practice drawing these out. You need to be able to do them from memory. Hot oxygen top right, carbons on the other corners, 4 bonds for all the carbons, hydrogen above, hydroxyl below, hydrogen above, hydroxyl below, hydrogen above, hydroxyl below. These 2 are different. Hydrogen below, hydroxyl above, hydrogen below, CH2OH above. Now, the way glucose molecules join together is something called a condensation reaction. Now, condensation reactions join molecules together, and in the process, they produce a molecule of water. Just like if you think about a condensation on your window at home, it's when you get water on the window. So, the first thing you need to be able to do is circle where the water will form. So we circle it like that. There's our molecule of water, and we can say plus H2O like that. Then what you do is you remove that because that's now in the water, and you should find that it leaves you with 1 oxygen. Now, in order to form the bond, we need to move that oxygen. So I'm going to rub it away, but it's still there. It's just that we move it into the middle and we draw the bond like that. And that bond is called a glycosidic bond. Oops, sorry. Glycosidic bond. And it's actually more specifically a 1,4 glycosidic bond, because remember how we label our carbons? 1, 2, 3, 4, 5, 6. And same here. 1, 2, 3, 4, 5, 6. Because it's going between carbon number 1 and carbon number 4, it's a 1,4 glycosidic bond. Okay. Now, there's a few different disaccharides that you need to know the names of. So the first one you need to know is maltose. The second one you need to know is sucrose. And the third one you need to know is lactose. Now, I have a little memory aid for 2 of these. I remember my great-grandmother for maltose, because M for maltose, G and G, because it's made of alpha glucose, G for glucose, and alpha glucose. In order to remember sucrose, I always think silly green frogs. So the S is for sucrose. The first G is for glucose, so it's made of alpha glucose. And the F for fructose, and fructose. And the third one you need to know is lactose. I don't have a memory aid for that. They're always made of glucose and something. These three you need to know. So it's made of alpha glucose. And the second monosaccharide that it's made of is galactose. I can just remember that because lactose is made of galactose. It's the same word. So all of these sugars are basically two sugars like that, two monosaccharides, and then these three disaccharides all have this structure where two of those are joined together. Okay, so two different monosaccharides form a disaccharide, and they're the three that we need to know. So the next type of carbohydrate that we need to know is a polysaccharide. So polysaccharides are made from lots of sugar units. So we could say lots of monosaccharides joined in the same way as we've looked at. So what kind of reaction is it? So we can say condensation reactions. And what type of bonds does it form? Glycosidic bonds. Okay, so polysaccharides are made of lots of monosaccharides by condensation reactions which form glycosidic bonds. Okay, now there's lots of different polysaccharides, and some of them are only in plants and some of them are only in animals. So we look at animals first because people are animals. So the way we store our glucose is a molecule called glycogen. And the way plants store their glucose is in a polysaccharide called starch. And we'll have a look at starch in a minute. So these are both ways of storing glucose. It's like energy storage. Now, plants also have another polysaccharide called cellulose. Cellulose isn't for storage. It's actually part of the cell walls. Plants have cell walls made of cellulose. So let's have a look. Ooh, excuse me. First of all, at the structure of glycogen, which is the way we store glucose in animals. So glycogen is a polysaccharide made of starch, okay, and basically it's lots of glucose molecules, lots of alpha glucose molecules joined together in a line. Now we've looked at how two alpha glucose molecules join, and it's exactly the same, but it goes on for many glucose molecules rather than just two. So we know already that these bonds here that join them together are called 1,4 glycosidic bonds. Now, glycogen isn't as simple as just a chain like this because it actually has branches as well. So rather than just having a straight chain of these molecules like this, we actually sometimes get a branch coming off. So you know how we have the CH2OH sticking up just there? We're just simplifying it, not drawing it every time. But what happens is joining onto that carbon, we get another chain joining on, going off like that. Now, often we wouldn't draw it like that. We would just draw it as a chain with a branch like that. Okay, we'd simplify it. But the important thing that we need to know here is about this bond just here. So we know that this carbon here is a 6, and this carbon here is a 1, because that would be our oxygen there. And we count around 1, 2, 3, 4, 5, 6. So that bond where we've got a branch is called a 1,6 glycosidic bond. So in glycogen, we've not just got 1,4 bonds, we've got 1,6. And whenever we got a branch like that on the molecule, that's where we're going to have a 1,6 glycosidic bond. Now, glycogen actually coils up. So rather than just being a straight chain as we've drawn it so far, it actually is a coil like this with branches coming off. Okay, and some of those branches might have branches coming off. So it's got lots and lots of branches in this coiled shape. So it's made of alpha glucose molecules joined in condensation reactions by glycosidic bonds. Along the chains, we've got 1,4 glycosidic bonds, and wherever we've got a branch, we've got a 1,6 glycosidic bond. Now, it has lots of branches, and we need to explain how these structural features have a function. So it has lots and lots of branches, and that means it actually has lots of ends. So if I get the highlighter tool here, we've got an end just here, an end just here, an end just here, just here, just here, just here, just here. So we've got lots and lots of ends. The more branches a molecule has, the more ends it has. And that means that it's got lots of places where we could remove a glucose molecule. And that means that we can remove glucose molecules very quickly. Why do we want to remove glucose molecules? Well, glucose is used in respiration to get energy. So we can say for respiration, for energy. So be careful that we're not talking really about glycogen storing energy. It does store energy, but be careful in your language in that really it stores glucose, and the glucose can be used in respiration to release energy. It's actually storing glucose. So having the branches is a benefit because it has lots of ends, so we can remove glucose really quickly in order to use it in respiration to get energy really efficiently. The other adaptation of glycogen is it's very compact. Again, be careful with your language. By being compact, we can say that it can store lots of glucose in a small space. Be careful that you don't say it stores lots of energy in a small space because they might pick you up on that in the mark scheme. So it can store lots of glucose in a small space. And then obviously the glucose could be used in respiration in order to release the energy from it. Okay. So that's your glycogen. Now, in plants, they store the energy in a different molecule. They store it in a molecule called starch. Now, starch is actually more complicated than glycogen because it's made of two components called amylose and amylopectin. So glycogen is just made of glycogen, but starch is made of amylose and amylopectin. It's made of two molecules. Amylose is very similar to glycogen in that it's alpha glucose joined by 1,4 glycosidic bonds in a chain, but it's just a single chain that goes into a helix. There's no branches. Amylopectin is similar to glycogen in it's this coiled chain of alpha glucose molecules. It actually has branches, so it has more ends than amylose, okay, but not as many branches as glycogen. So you could say that amylopectin stores, or we're able to release the energy from amylopectin more quickly than amylose because it's got more ends, because it's got branches. However, you can't release the glucose as quickly as you could in glycogen because it's not got as many branches and therefore not got as many ends. Okay, we do not have starch in people. We do not store our glucose as starch. That's just in plants. Okay. If you're thinking about the bonds involved, amylose has only got a single chain, so it's only going to have 1,4 glycosidic bonds. Amylopectin is going to have 1,4 glycosidic bonds, but at the branches where the branches join on, you're also going to have 1,6 glycosidic bonds, like we had in glycogen. Okay. In addition to starch, we don't need to really know much about it, but plants also can put the glucose molecules together into a molecule called cellulose. Now, cellulose, for your information, it's not in your textbook or anything, but just for your information, is not a coil. They're straight chains, and they've just got these cross links between the chains. And the difference is that it's made of beta glucose, not alpha glucose. So there is a little bit different to the energy storage molecules. And we find that in plants in the cell walls. Their cell walls are made of cellulose. Right, now the last thing that we need to know about carbohydrates is a process called glycogenolysis. It's glycogenolysis. So, but really, if we break it down, it's glycogen lysis. So we've already looked at glycogen as a molecule. That's the polysaccharide that animals store their glucose as. And the word lysis means to break up. So glycogenolysis is breaking down the glycogen. So we know that glycogen is how glucose is stored. And basically, it's just lots of glucose molecules joined together. Lysis means to break something down. So when we break down glycogen, it's called glycogenolysis. Okay, so think about the glycogen molecule, this coiled structure. It's a chain of alpha glucose molecules, and it's got lots of ends. When glycogenolysis happens, we just break a glucose molecule off one of these ends. Then we can use that glucose molecule in respiration to release the energy. Now, the next thing is, when would your body need to do this process? Now, your body is always monitoring the glucose levels in your blood. And when your blood glucose level goes too low, we need to get more glucose. So what happens is that this stored glucose in these glycogen molecules needs to be released. So when blood glucose levels drop, glycogenolysis occurs. We start to break down the glycogen to release the glucose. So blood glucose too low, then we get glycogenolysis, which releases the glucose from that polysaccharide. Okay. Now, there is an enzyme that does this. So in order to break these glycosidic bonds, these 1,4-glycosidic bonds, to release one of these little glucose molecules from the end of this polysaccharide, the enzyme that actually does that is called glycogen, because we're breaking down glycogen, phosphorylase. That's a lot of detail to remember, but if you're ready for more detail, that's the name of the enzyme that breaks that bond in order to release the glucose. Okay. Then it actually needs a second enzyme, which is called a glycogen debranching enzyme, if it needs to break the bond at the branch point. Okay, so where a branch joins on, it needs a different enzyme, which is called a glycogen debranching enzyme. Okay, most of you are just going to know that enzymes are used to break the bonds to release the glucose molecules, but there's the names of some of the enzymes involved if you want to use those words as well. Okay, so the idea of glycogenolysis as an overview is that it's breaking down the glycogen to release the glucose molecules, so they can then be used in respiration to release the energy that's stored in them. And that's the end of that video.